Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. Today, we're going to further discuss lactate dehydrogenase as a potential therapeutic target as a way to enhance mitochondrial metabolic therapies. In particular, today, we're going to be looking at drugs and which of those drugs have an effect on this very important enzyme. So without further ado, let's get into it. So in the last video, we talked about LDH inhibition, but from more of a mechanistic perspective. If we inhibit this enzyme, what functionally happens to the cancer cells? And now today, we're going to be talking about in particular which drugs show an effect on lactate dehydrogenase. So the first paper I'm going to pull was very recently published in July of 2025, and it says lactate dehydrogenase a potential new target for tumor drug resistance intervention. It says here, tumor drug resistance is a crucial scientific and technological issue that urgently needs to be addressed in cancer treatment. In recent years, research on lactate dehydrogenase A, LDHA, in the field of tumor drug resistance has progressively deepened with breakthrough advancements achieved. Due to their metabolic characteristics, tumors often exhibit abnormal LDHA expression, which meets their growth requirements by promoting glycolysis and lactate production. A series of studies in recent years has revealed that in drug-resistant tumors, LDHA regulates its own expression through molecules such as transcription factors and non-coding RNAs, and regulates tumor drug resistance by influencing the stemness of tumor stem cells, the tumor microenvironment, the TME, and intrinsic tumor drug resistance. In this paper, we first present timely updates on the expression and regulatory mechanisms of LDHA. Importantly, we systemically expound on the mechanisms through which LDHA is implicated in drug resistance to radiotherapy, chemotherapy, and immunotherapy in tumors, including lung and breast cancers. We further discuss a series of scientific issues that urgently need to be elucidated in further research in this field, aiming to provide more evidence and references for elucidating the mechanisms of LDHA-mediated drug resistance and developing new intervention strategies against tumor drug resistance. And I'm going to pull us back to the graphic that we saw, this figure 3 or table 3, as part of the paper that we covered in the last video. And in particular, there is one molecule on this list that seems to be the most studied of all of them, and that would be oxamate. And so the first paper I'm going to talk about when looking at oxamate was published in March of 2022 and is titled Oxamate Targeting Aggressive Cancers with Special Emphasis to Brain Tumors. And it says here, cancer is one of the main causes of human mortality, and brain tumors, including invasive pituitary adenomas, medulloblastomas, and glioblastomas, are common brain malignancies with poor prognosis. Therefore, the development of innovative management strategies for refractory cancers and brain tumors is important. In states of mitochondrial dysfunction commonly encountered in malignant cells, cells mostly shift to anaerobic glycolysis by increasing the expression of lactate dehydrogenase A gene. Oxamate, an isosteric form of pyruvate, blocks LDHA activity by competing with pyruvate. By blocking LDHA, it inhibits pro-tumorigenic cascades and induces reactive oxygen species, ROS, induce mitochondrial apoptosis of cancer cells. In preclinical studies, oxamate blocked the growth of invasive pituitary adenomas, medulloblastomas, and glioblastomas. Oxamate also increases temozolomide and radiotherapy sensitivity of glioblastomas. Oxamate is highly polar, which may preclude its clinical utilization due to low penetrance through cell membranes. However, this obstacle could be overcome with nanoliposomes. Moreover, different oxamate analogs were developed which can inhibit LDHC4, an enzyme also involved in cancer progression and germ cell physiology. Lastly, phenotropin, an anti-diabetic agent, exerts anti-cancer effects via complex 1 inhibition in the mitochondria and leading to overproduction of ROS. Oxamate combination with phenotropin reduces the lactic acid causing side effects of phenotropin while inducing synergistic anti-cancer efficacy. In sum, oxamate as a single agent and more efficiently with phenotropin has high potential to slow the progression of aggressive cancers with special emphasis to brain tumors. And basically what we have here is a graphic that is somewhat similar to what we've seen in the past, where we have pyruvate not being able to be converted, right, to lactate because lactate dehydrogenase A is being inhibited by, in this case, oxamate. But we see a bunch of other like side benefits that oxamate could potentially provide in the way of inflammation reduction, cell cycle, increased ROS, and mitochondrial apoptosis in cancer cells. And then lactate, we know, is what leads to increased cancer cell growth, invasiveness, impaired host immunity and surveillance, enhanced angiogenesis and metastases. So therefore, by cutting that off, we are then reversing these processes that are happening. I think you can see how this particular enzyme system could really break up some of these vicious cycles that we've seen over and over again. The next paper is titled LDHA inhibitor as a remedy to potentiate the anti-cancer effects of docetaxel in prostate cancer, and it was published in January of 2024. And basically what it says is that reprogramming glucose metabolism with an LDH inhibitor can increase the effectiveness of docetaxel on apoptosis and metastasis mechanisms in prostate cancer. And we have basically where the injections of several substances, whether it be the control of prostate cancer or docetaxel or an inhibitor by itself or the inhibitor plus docetaxel. So we see here that this is the first day of the injections at day 20. They basically allow the cancer to just grow rampantly until then. They inject it with whatever substance it is. And if you just let it go, right, it's going to just keep growing. If you give it either the chemotherapeutic agent docetaxel, which is the blue, you see it start to go down after about 5 days. And then the green actually has a more profound effect by itself, even earlier, this LDHA inhibitor. And then if you combine them, you see a synergy that's happening there too. So this is just one illustration of how an LDHA inhibitor could enhance the standard of care in the form of chemotherapy, and in this case, docetaxel. The next paper we're going to look at was published in March of 2023 and is titled The Rationale for LDH Targeted Cancer Immunotherapy. And it says here that immunotherapies have significantly improved the survival of patients in many cancers over the last decade. However, primary and secondary resistances are encountered in most patients. Unraveling resistance mechanisms to cancer immunotherapies is an area of active investigation. Elevated levels of circulating enzyme lactate dehydrogenase, LDH, has been historically considered in oncology as a marker of bad prognosis, usually attributed to elevated tumor burden and cancer metabolism. Recent evidence suggests that elevated LDH levels could be independent from tumor burden and contain a negative predictive value, which could help in guiding treatment strategies in immuno-oncology. In this review, we decipher the rationale supporting the potential of LDH-targeted therapeutic strategies to tackle the direct immunous effects of LDH on a wide range of immune cells and enhance the survival of patients treated with cancer immunotherapies. I just want to, show that this paper says that you're going to likely encounter immunotherapy resistance in most patients treated with these drugs. So if you are getting onto a clinical trial where immunotherapy is involved, just know that that is not going to be a long-term strategy for you likely. And therefore, I'm not saying you should do it. That's between you and your oncologist and your family. But my point is that you should be thinking about ways, in my opinion, about how to overcome this known problem that happens. And one of the ways is looking at LDH. But what does LDH functionally do? It shuts down the lactate dehydrogenase enzyme, yes. But what it functionally does is it puts a brake on aerobic glycolysis, which is the big problem that leads to chemotherapy resistance and immunotherapy resistance, because lactate, the end product of that system, is what leads to this immune suppression and failure of chemo and immunotherapies. So therefore, really, if you break the cycle upon any part of this pathway, then you likely are enhancing the standard of care. Just want to put that out there. So, introduction. Over the past decade, immune checkpoint blockades, ICBs, a wide range of metastatic tumor types, a broad spectrum of ICB agents have been approved, such as antagonistic antibodies against program death receptor 1, PD1, program death ligand 1, PDL1, and cytotoxic T lymphocyte associated 4, CTLA4. One of the biggest challenges is to unravel the tumor escape mechanisms limiting the ICB or the immune checkpoint blockade efficacy, since the majority of patients will either not benefit from those treatments, primary resistance, or relapse after a period of response, secondary resistance. And what we see here, and this is again a different nice graphic specifically looking at immunity and how lactate or lactic acid that's being secreted by these tumor cells is what's responsible for the anti-tumor immunity and the immune evasion. So this is the problem, right? Lactate is the problem. The drug itself is not the problem. The problem is the lactate. So what they're saying is, well, if we inhibit, since we know this is the problem, if we inhibit LDH, would that not enhance anti-tumor immunity and lead to less immune escape and allow our drugs to work better? Yes. So what they're saying is, let's block LDH. Whereas what I'm saying is, if you block glycolysis anywhere along that pathway, I guarantee you the same process will likely happen. You're going to have improvements in these therapies. And we've seen that. Now we're at the end of glycolysis. We're at the functional end of glycolysis. We have now went from pyruvate, the end of glycolysis, to now the fermentation of glucose because the mitochondria are damaged, right? So every enzyme system that we looked at, whether it be hexokinase, whether it be phosphofructokinase, whether it be pyruvate kinase M2, all of these enzymes that we've looked at show a synergy with the standard of care. And the reason is because they're ultimately at different levels of that process, leading to less lactate that is involved. And lactate and succinate are the issues of leading to this anti-tumor immunity blockade. Early phase 1 and 2 clinical trials testing inhibition of LDH shows promising results in certain cancer types warranted confirmation in randomized controlled clinical trials. Furthermore, research on systemic and intratumoral biomarkers of immunological response following these novel treatment strategies can aid in the development and application of combination therapeutic strategies of cancer immunology. Ultimately, improved understanding of the temporal complexity of cancer metabolism and prognosis in association with immune escape is crucial to the correct timing and combinations of treatments. Conclusion. One of the hallmarks of cancer cells is the metabolic switch to aerobic glycolysis, with LDH representing a key enzyme in this process. Circulating LDH levels are high in many advanced cancer patients. While increasing levels of LDH can be considered a consequence of cancer cell metabolism, it is also a cause of many inherently tied hallmarks of cancer, including angiogenesis, invasion, metastases, and immune evasion. LDH inhibition has been deemed relatively safe and has demonstrated promising results experimentally in the context of immunotherapy combinations and preclinical models. Because there is a clear mechanistic association between LDH elevation and poor survival in cancer, inhibiting LDH could improve patient outcome and make immunotherapies more effective in various types of cancer. However, there is a need for more investigations to determine, apart from their safety and efficacy, which LDH isotype should be targeted, how and which patients should be treated with LDH inhibitors. Understanding the specificity of LDH changes in the context of anti-tumor response can guide the design of selective therapeutic strategies. Importantly, the level of evidence accumulated over the recent years on the negative impact of high circulating levels of LDH across cancer should make mandatory the stratification of cancer immunotherapy clinical trials on this easily implementable biomarker. That's actually a really good point. And it's something that I've been meaning to say is basically that this is not like a pie in the sky lab marker, right? This is not something that like only research labs have access to. I can order this easily all the time. Most of the time, frankly, the blood that's been ordered in the ER, like the CBC or CMP, I can add it on. I don't have to even stick the patient again. I can just add it on to the labs. It's already been drawn. So this is something that is readily available and it's fast, it's easy, it's cheap. And what they're saying here basically is that LDH levels should help maybe guide the cancer immunotherapy clinical trials, because if you have high levels of LDH, which is easily detectable as a blood test, you're going to have likely immunotherapy failure or at least like poor responses. And that could guide whether or not an LDH inhibitor should be added. Now, I personally think that's a really backwards way of looking at it. I think that you should be looking at blocking all of the substrate level phosphorylation, both from a glucose side and a glutamine side first, and then adding things if it's necessary. But of course, they're looking at it from the perspective that the drug is the primary intervention, and this is a way to augment it or as an adjuvant. But the bottom line is LDH is an important biomarker and something we can actually check. Unlike a lot of these other enzyme systems like hexokinase or phosphofructokinase or glucose transporters or pyruvate kinase M2, we can't functionally check for those clinically, but LDH is one that we can check easily. And I think that this is what makes it, for me, a very special enzyme. Not necessarily that blocking LDH does something significantly better than blocking hexokinase per se, because we're blocking the same system. We're blocking glycolysis of the fermentation of glucose into lactate. But because we can use this clinically as a way to track to see where you're at on that spectrum of kind of like aggressive disease and malignancy and prognosis, I think it kind of makes it more real in some ways because of that. The next paper we're going to look at is from April of 2021 and is titled Targeting Lactate Dehydrogenase A Improves Radiotherapy Efficacy in Non-Small Cell Lung Cancer from Bedside to Bench. And it says here that both mRNA and protein levels of lactate dehydrogenase expression were upregulated in non-small cell lung cancer tissues. High LDHA expression was a poor prognostic factor and associated with radioresistance in these non-small cell lung cancer patients. LDHA inhibition by oxamate remarkably increases radiosensitivity in both these A549 and H1975 cancer cells, and enhanced ionizing radiation, IR, induced apoptosis and autophagy, accompanied by cell cycle distribution alterations. Furthermore, LDHA inhibition induced reactive oxygen species accumulation and cellular ATP depletion, which might increase DNA injury and hinder DNA repair activity. Our study suggests that inhibition of LDHA, lactate dehydrogenase A, may be a potential strategy to improve radiotherapy efficacy in non-small cell lung cancer patients, which needs to be further tested by clinical trials. So So we're using as the lactate dehydrogenase A inhibitor oxamate, signified by this OXA. Oxamate is inhibiting LDHA, which is going to block glycolysis, and that's going to lead to mitochondrial OXPHOS kind of activation with a broken mitochondria, which leads to exhaust or reactive oxygen species that are put off. That's going to damage DNA, and it's going to lead to the ejection of cytochrome C, which is going to lead to apoptosis, and it's going to make just basically these cancer cells more susceptible to radiation therapy. This paper is titled "Targeting Lactate Dehydrogenase A Exerts Anti-Leukemic Effects on T-Cell Acute Lymphoblastic Leukemia," published in October of 2020. And the reason that I particularly pulled this paper is not that it really shows us anything per se more surprising or more groundbreaking in terms of using oxamate to block lactate dehydrogenase A, but it's more the fact that most of the papers that we see employing a metabolic approach to cancer, whether it's trying to improve the standard of care through chemotherapy or radiotherapy, or just providing proof of concept, most of those studies are done in solid tumors. And whenever I can find a paper that kind of like highlights the effect happening in non-solid tumors also, I like to do it, because a lot of the people who have non-solid tumors believe that they are kind of exempt from these metabolic therapies, which is not true, but nonetheless, they kind of feel that way. And, that's why I, for example, like to show slides like this, right? Because Seifried did a great job, and his group did a great job of showing how mitochondrial dysfunction is noted in all of these cancers, right? But still, the awareness doesn't necessarily seep through to the people who have non-solid tumors. And therefore, I like to give a special kind of shout out to those who have those tumors, because I want to let them know that this also applies to you and can be likely effective, God willing, for you as well. So what it says here is that oxamate significantly inhibited proliferation and induced apoptosis of JERCAT and DU528 cells. It also arrested JERCAT and DU528 cells in the G0, G1 phase and simulated ROS production. Blocking LDHA significantly decreased the gene and protein expression of c-Myc, as well as levels of phosphorylated serine threonine kinase, AKT, and glycogen synthase 3 beta, and phosphatidylinositol 3 kinase PI3K signaling pathway. LDH gene knockout delayed the disease progression and downregulated c-Myc RNA and protein expression in all in TALL transgenic zebrafish. Targeting LDHA exerted an anti-leukemic effect on T-cell acute lymphoblastic leukemia, representing a potential strategy for TALL treatment. So most people have never heard of oxamate. And like it said in one of the research papers, that oxamate has kind of a poor lipophilic absorption, so it's not able to cross membranes very well, which may kind of limit its clinical ability unless it's kind of altered chemically, and that's kind of like in drug development. So we're not there yet, basically, right? So what we have is a proof of concept. We have that when you block lactate dehydrogenase A, there is going to be improvements in antitumor immunity. It's going to enhance the standard of care, chemotherapy, radiotherapy, and immunotherapy. We saw examples of all three of those happening with the use of oxamate. But what is not shown, again, you have graphics like this in these papers, and you think, well, these guys are researchers in this particular subject of medicine. They must know all of the exhaustive lists of things that potentially have effects on lactate dehydrogenase A. And that's unfortunately not the case, which is why you have to do your due diligence. And so therefore, I was able to find this paper, which is fairly recent. It was published in September of 2020, and it's titled "Low Glucose Enhanced Metformin's Inhibitory Effects on Pancreatic Cancer Cells by Suppressing Glycolysis and Inducing Energy Stress via Upregulation of this MIR-210-5P." And what it says here is that metformin significantly inhibited the expression of this PFK-FB2 in the low glucose group and inhibited the LDH activity both in the low and high glucose group, thus inhibiting aerobic glycolysis and inducing energy stress. Cells in the high glucose group could make a compensatory adaptation to the energy stress induced by metformin through increasing glucose consumption. However, due to the limited glucose supply and high dependence on aerobic glycolysis, the cell in the low glucose group, they couldn't make an effective adaptation compensation. Therefore, cells in the low glucose group were more vulnerable to the toxicity of metformin. So a couple interesting things were found in this paper. They split this up into a high glucose group and a low glucose glucose group, number one. So basically, someone who is eating whatever they want, because eating whatever you want doesn't affect cancer, right? And so they have the high glucose group, and then they have someone who maybe has listened to Seafried and is on a ketogenic diet. And so we have two different groups of people. Both of them are getting metformin. Interestingly enough, metformin blocks LDH. Again, not seen on graphics otherwise or tables otherwise, but it has an effect at inhibiting LDH. And so when metformin was given to people who didn't change their diet or lifestyle, right, the quote unquote high glucose group, representing basically the high glucose group, that would be able to be overcome, even though you're blocking a key enzyme here, lactate dehydrogenase. Because glucose was so available, it was able to figure out a way to get around that blockade. However, if they were looking at the low glucose group, which is basically representing a population that would be on a low carbohydrate or a ketogenic diet, then when they gave metformin, the blockade was too strong for the cancer cell to be able to overcome that, because there's less glucose available in general to kind of find a way around it. So I think this illustrates a couple of points. Number one, it illustrates the point that just being on a ketogenic diet, for example, or just using a drug that blocks a certain enzyme system is likely not the solution. We have to look at this as a system. So that's why when we look at graphics or we look at papers that Seafried and his group have published, you have the presses, which is basically the low glucose, right? And then you add glucose inhibitors and glutamine inhibitors as a way to further lead to its demise. If you were just to take the inhibitors and be on a high glucose kind of diet, or not be willing to change your diet to be at least trying to mitigate your glucose level, then you're likely not going to have success. The other thing I would say is that it also kind of illustrates that you can't hang your hat on one particular enzyme to be blocking at a time. So there is a lot of promise with every single enzyme that we've looked at, whether it be hexokinase, whether it be a glucose transporter, whether it be phosphofructokinase, whatever. And they're going to say, oh, this is a game changer. Like this needs to be looked at clinical trials. This is a promising thing. But I'm telling you, if you look at it very siloed like that, I think that that's where you're going to basically be in the same boat that medicine is with the biologic whack-a-mole. I think that it's going to have to be looked at from multiple levels, looking at things that block glucose uptake, looking at things that block hexokinase, phosphofructokinase, pyruvate kinase, lactate dehydrogenase, as well as being on a ketogenic diet. It needs to be a system of therapies that work together upon the same axis so that you can minimize having a way around what we're trying to do functionally. And that's really well illustrated in this paper, just by something as simple as having a low glucose group and a high glucose group showing a different effect of metformin. That by itself shows you that you can't just eat whatever you want with cancer and you're going to have the same effect. This is not functionally true. It doesn't make any sense. So that's why the foundation of ketogenic metabolic therapy is a low glucose diet, right? It's not the only reason. There's insulin, there's ketones, there's other things that happen when you're on a low glucose or a ketogenic diet. But you can see here, and this is a paper, that where metformin goes from being ineffective or less effective to highly effective when the conditions or the environment is different. And I thought that was very interesting. And I wanted to really highlight this particular paper. Not only the fact that I couldn't find metformin on any graphic anywhere else until I did this search. What else do we find when we look at the search? Well, what about ivermectin? This is an oldie but goodie, isn't it? December of 1994, this paper was published. Effective ivermectin on the activity of enzymes in the mammalian cells in vitro. And what does that say? At 72 hours time course following ivermectin exposure indicated a decrease in the activity of lactate dehydrogenase. Imagine that. What else do we find? From September of 2023, mebendazole targets essential proteins and glucose metabolism, leading to gastric cancer cells to death. And what did it say? Results evidenced that the antitumor and cytotoxic mechanism of mebendazole in gastric cancer cells is related to the modulation of RNA, mRNA, expression of glycolytic targets, SLC2A1, which is a glucose transporter, hexokinase, GAPDH, and lactate dehydrogenase A. Moreover, in silico analysis has shown that these genes are overexpressed in gastric cancer samples, not to be surprised, and this increase in expression is related to the decreased overall survival times. Molecular docking revealed that mebendazole modifies the protein structure of these targets, which may lead to the changes in their protein function. In vitro studies also showed that mebendazole induces alteration of glucose uptake, LDH's activity, lactate dehydrogenase enzymatic activity, and ATP production. So in this search, we found that oxamate is kind of the prototypic LDHA inhibitor. But we also found in our search that is maybe not that clinically relevant because number one, it's probably not going to be able to be available outside of chemical research stores. And on top of that, it probably doesn't have good biologic activity at getting to where it needs to go. So therefore, it limits its clinical availability. But we know that when we block LDH, it enhances all these other therapies, right? And it totally makes sense too, because it's completely working on the cytosolic substrate level phosphorylation or the Warburg effect. But then in our search, we found that metformin also has an effect here, ivermectin has an effect here, and mebendazole has an effect here, which all tie into how we're already potentially approaching ketogenic metabolic therapy as a whole. So therefore, we can see how these could dovetail nicely into our overall strategy. So we now have looked at LDH inhibitors, the mechanism of how they functionally work and how they lead to cancer cell death. We've now looked at drug inhibitors. And next, we will go into the natural available inhibitors of lactate dehydrogenase in the next video. If you like videos like this, please like, share, and subscribe. Until next time.